it's good to know, because chances are you, a loved one, or a friend are going to get cancer. It's above 25%. Okay, 25% chance or above that someone gets cancer. Okay, so it's something that is critical. Now, as I mentioned, we spend our entire lives riding this line of cell growth and cell death. Cell death is critical to shape our organs, our tissues, as well as get rid of problematic cells, such as cancer. But we can't give too much cell death, because then organs start failing and organs start dying because we can't regenerate our tissues. So we ride this line all of our lives. Now, as we undergo life, we're exposed to a number of different things in our environment that increase our chances of developing cancer. We call them carcinogens. They're anything from UV radiation. Most of the time we're not exposed to gamma radiation, but X-rays. X-rays can increase your chance of getting cancer. That's why they usually shield your body when you have X-rays done in your teeth or in different parts if you've got a broken bone or whatnot, just to minimize the exposure of your other cells to X-rays, because they'll mutate your DNA and increase your chances of having cancer. So we're going to talk about what ultimately causes cancer. This is why we talked about the cell cycle, because that's what cancer is, a disease of the cell cycle. The cell regulates its growth, where it says, oh, I need to divide every 24 hours. That's what your skin does. So the stem cells in your skin are saying, oh, clock's ready, okay, we've replicated all of our DNA, time to divide. And it just goes through this every 24 hours. What controls the cell cycle are a series of genes, just like genes that make your hair a certain color and your blood type a certain type and whatnot. There are also genes that regulate how fast the cells grow. So, like any gene, it can be mutated. You can change the nucleic acid sequence due to some type of radiation, due to some type of chemical, like eating a Twinkie or something, There are some foods that have carcinogens in them. They found that a lot of the artificial sweeteners and other things that we use are highly mutagenic, where they have a high risk of causing your DNA to mutate. Chemicals that you may work in gardening, there's a lot of things that we're now beginning to understand that when they get absorbed into our skin and get into our blood and whatnot, have a higher rate and chance of causing the cells to become cancerous. Now, there's always just your predisposition based upon the genetics you've gotten from your parents. And at the very end of this lecture, we'll talk about how you can inherit genes that give you a predisposition to cancer. Now, they don't give you cancer, but they make you more disposed to developing cancer sometime in your lifetime. All right. Now, with the few exceptions, most of the cells in your body are dividing. Some occur very rapidly, divide very rapidly, others not so rapidly. And there are ways to increase this growth rate. For example, you cut your skin. Well, if it only grew every 24 hours after you cut your skin, it would take forever to heal. So there are usually hormones that can be secreted by tissues that stimulate growth. So you can artificially accelerate cell growth, and you can retard it or cause it to slow down. So there are growth factors that your body releases, especially when teenagers go through growth spurts. That's what's going on, is you're exposed to these growth hormones and these growth factors that are increasing your bone growth, increasing all of these changes that ultimately come down to protein stimulation. Now, what are they actually stimulating? They're stimulating genes that regulate the cell cycle. Okay? So, this is how our body works. We essentially, when we need to accelerate cell growth, we'll usually have some type of signal. Like, you damage your skin, and the cells are like, oh, and they release a signal to say, hey, you need to grow faster to repair the damage to the tissue so that we can do what we're supposed to be doing, protecting the underlying organs and things of that sort. So there are certain key stop points Now, these aren't the only ones there are, but these are just a couple of examples of some of these stop points. These are checkpoints that you have to go through. And if you don't have the right papers, so to speak, meaning you don't have the ability to go past this point, then the cell will usually trigger apoptosis. Because if you don't pass one of these checkpoints, then there's usually some problem with the cell. Now, a lot of times, some of us may have gotten cancer and never even realized it because the default kill yourself cell mechanism, apoptosis, got triggered. And therefore, the cells never became cancerous because they killed themselves. But there becomes a problem when they ignore these checkpoints and just run right through them. So what are some of these checkpoints? For example, the first one here is a proofreading of your DNA. If your DNA gets damaged so much that the cell's like, you know what, just screw it. I can't even make all these repairs. If it's damaged beyond repair, this will induce apoptosis. In fact, we'll show that this is exactly what radiation therapy essentially does. Radiation therapy is meant to cause the cells to induce apoptosis by not letting them pass this checkpoint. So there's a protein. In fact, this is one of the most commonly mutated proteins that gets inherited in the human species that gives you a predisposition to cancer. Okay? That gene right there that does that checking. Now, there's other checkpoints. After, let's say you're like, you know what, the DNA's good. We don't need to repair it too much. So it'll copy all the DNA. Because it's not going to try to copy the DNA if it's just damaged beyond repair. So it copies all the DNA. Well, it won't begin mitosis until it's sure that the DNA has been done replicating. There's no point in going into mitosis unless it's all been replicated and been replicated correctly. So it checks it and makes sure and readjusts tiny little errors. That's why usually when you go through this process, there's only about 3 mistakes that get overlooked. 3 in 3 billion. Okay? Those are good odds. So it also needs to make sure that everything's ready for mitosis. If it's not, then it stops. Now, if it stops too long at this stage, then it will say, you know what, screw it, something's wrong, and it'll kill itself. So there's lots of checkpoints in where the cell can be killed. Now, cancer typically comes about when the cell ignores a lot of those checkpoints and just grows no matter what. It's like, you know what, screw it. I'm just going to keep going. I'm going to keep replicating. I don't care what the cell's telling me to do. And that's a problem, obviously, because one of the biggest issues with cancer is that it ultimately robs your tissues and your cells and your organs of its nutrients that it needs and causes stroke in your brain or cell death in the organs and causes organs to fail and whatnot. Causes blood clots because it clogs up, various vessels and things of that sort. So too many cells forming these masses in various organs is ultimately what causes all the problems. Now, the first thing I'm going to test you on is how we classify cancer. Now, these are not all of the types of cancers. This is just a small little sampling of a few names of some well-known and not so well-known cancers. Now, generally speaking, carcinoma is essentially a cancer of the organ, but we usually add the name of the organ to it. So, for example, pancreatic carcinoma is a cancer of the pancreas. Or you could say basal cell carcinoma. This is one of the most common types of skin cancer. This is the skin cancer that usually occurs through too much sun tanning and the like. Okay? So carcinoma by itself really doesn't tell you where the cancer is. It's just cancer of the organ. We usually quantify it by saying pancreatic carcinoma or basal cell carcinoma or whatnot. Now, some cancers have very specific names. For example, if it's in the muscles, we call it a sarcoma. Okay, so muscle cancer is sarcoma. Leukemia. Everybody's heard of leukemia. This is cancer in the blood. Okay, so this is when you typically have white blood cells that are just growing out of control. Melanomas. This is another type of skin cancer. Now, a lot of times the cancer can be classified either by the organ or by the cell type. So, let's look at this. there is a type of cell in your skin called melanocytes. Now these melanocytes, do a quick anatomy here. You've got the dermis of your skin, which is the main layer. You've got the epidermis, which is the top layer. The two types of skin cancers as follows. Basal cell carcinoma has to do with the stem cells that regenerate the epidermis. These are the cells that grow once a day. Okay? These are the cells that typically are regenerating and renewing your skin once a day, layer by layer. Okay? When these get mutated and start to form tumors, they're very apparent. That tumor mass is very apparent on the surface of your skin, which is why most skin cancers are 98% curable, because you just remove the tumor mass and no harm, no foul. However, melanomas are a little more problematic because they can go undetected for long periods of time. So where are the melanocytes? Well, they sit deep in your dermis, and they cause these little projections to go up into your epidermis. So what do? They secrete a protein called melanin. What's melanin? Well, it's a protein that has a very specific tertiary structure that absorbs UV radiation. And this is why you tan when you suntan, or you go into a tanning bed or whatnot. Your body is actually increasing the rate of melanin production to say, stop exposing me to UV radiation, because the UV radiation will damage your DNA. Your body has to repair the DNA every time it's exposed to UV light. UV light is very problematic to genetic material. So the more you're exposed to UV light, the more melanin your body will produce. Melanomas are when the melanocytes start growing out of control. Now these are usually seen by having dark patches in your skin. Sometimes they can protrude, but a lot of times they don't. Now, moles are not cancer. But if a mole is growing and continues to grow, usually a doctor will remove it, because it is abnormal cell growth of the melanomas. Of the melanocytes. Okay, so not all moles are cancerous, but they can become cancerous. Now, freckles are typically overactive melanocytes, but it has nothing to do with cancer. Okay, so freckles typically are high concentrations of melanin in spots in your cheeks and whatnot. And so that's kind of the relationship between some of these things. So usually moles, you're like, whatever. They don't grow much beyond a certain point. But if they start growing later on in your life, usually the doctor will go in and surgically remove it. But they have to get deep into the dermis to be able to do so. All right. So this is how we name them. Now there's other ones that you're going to see showing up later on. I think I also bring up a brain cancer called astrocytoma. So an astrocytoma comes from astrocytes, which are nerve cells in the brain. Usually these are the support cells that support a lot of the neurons. And those become very problematic, because not that you can't detect the cancer, is that many times you can't go in and remove the cancer without doing more damage or causing too much brain damage of the individual. So that's a big issue that we have to deal with, is removing the cancer, especially in organs that tear easily or usually go unnoticed for long periods of time. Naming it is important to understand this next concept, because there are two main types of tumors. Now, a tumor is essentially the overgrown mass of cells that come about when the cells lose control of the cell cycle. So when you get more cell growth than cell death, and the cells ultimately start forming this mass, that's what we call a tumor. Now, in the earliest stages of most cancer, you end up what's called having a benign tumor. Now, it doesn't mean a benign can't be life-threatening, because in fact, brain tumors that are benign, that are slow growing, can cause intracranial pressure and stroke and death. So just because it's benign doesn't mean that it's not life-threatening. However, on the whole, benign tumors are typically slow-growing and not as harmful. So that's what we mean by harmless, is that they're usually not a threat to your body's homeostasis at first. Okay? This also is encapsulated. Benign tumors typically remain in their site of origin. So if they're forming in your skin, then they stay in your skin. If they're forming in your liver, they stay in your liver or your bone or your muscle. Okay? Or breast cancer or testicular cancer, whatever the case may be. That's where you're checking for lumps. That's where you're checking for abnormal growth. Okay? If the tumor progresses because it goes unchecked or undetected or whatever the case may be, it can ultimately undergo a process called metastasis. This is when you start having big problems. A malignant tumor is one that has metastasized. So what does that mean? It means that cells have broken free from the encapsulated tumor mass and entered one of two main thoroughways in your body, either your cardiovascular system or your lymphatic system. These are the two major fluid systems in your body that transport things all through the rest of the body. So if cells go through those fluid systems, it can seed new tumors in other parts of your body, where you end up having skin cancer all of a sudden in your lungs, or you're having any number of issues where these cells are showing up in other organs, causing new tumors to form, and this is when it becomes just very, very difficult to treat. Okay, now let's look at how we treat cancer. These are many of the tried and true methods that we've used, although there are many that are being advanced today. These are in no way the best type of treatments. In fact, we're trying to get away from some of these because of how much harm they cause the body. But I'll describe what they are, why we still use them, and maybe some of the new developing things that we're coming up with. Now, most cancer research today is not necessarily going into developing drugs to kill cancer. It's in how to understand what's causing the cancer. That in and of itself is more important now than ever, to know what's causing it, because the more you know about what's causing it, the more you can target that type of cancer. Back in the day, we would just kind of use a shotgun approach of chemotherapy and radiation therapy, and sometimes it would work and sometimes it wouldn't. And you have to ask the question, well, why? Well, we'll answer that. Ultimately coming down to the reasons why two people could have leukemia, radiation works for one but doesn't work for the other. Well, why? So let's go over how this works. Now, as in almost all cases, when you have cancer, there always has to be some type of surgery to remove the tumor mass of cells. Not always. In some cases where you have something like leukemia, where it's already going through your cardiovascular system, you may not necessarily have a tumor mass. But in most cases, surgery is one of those things that just goes hand in hand, where you try to physically remove the mass of cells from the organ. In some cases, however, this is not possible without destroying the organ, be it a brain, be it the pancreas. Usually pancreatic cancers are so fatal because they go unnoticed for so long. And by the time that you see that it's shown up, it's almost too late. So it's not that we can't necessarily go in and remove it, but a lot of times they just go undetected for a long period of time. Now, radiation therapy. Here's the interesting thing. Radiation not only causes cancer, but is used to cure cancer. So, what's the trade-off here? Well, constant exposure to UV radiation, X-rays, any high-level radiation over time damages your DNA to the point where if you damage the cells that control the cell cycle, then the cells will start growing out of control. You get cancer. Well, what radiation therapy does are usually high, quick bursts of radiation to the cancer cells to damage them so badly that they cannot replicate, that they cannot undergo cell division, and it induces apoptosis. Remember I told you about that checkpoint that cells typically use right here? That's what radiation therapy is designed to do. It's designed so that when this gene comes in here and says, oh man, all the DNA is just screwed up, it will cause the cells to die. The problem with this is it also kills healthy cells. Now, what are the healthy cells that typically die? It's usually the most rapidly dividing cells in your body. Your hair, your immune system, the lining of your gut, and whatnot. And this is where you get sick and you have those problems, is because the most rapidly dividing your cells in your body are also triggered to undergo apoptosis. So there's this trade-off where you have to give enough radiation therapy to kill the cancer in doses and let the body recover in between. Hopefully the body recovers faster than the cancer recovers, and ultimately after several treatments you eventually eradicate the cancer without killing the individual. That's the trade-off. That's the kind of the difficulty of this. But not all cancers respond to radiation therapy, because a good number of them have a mutation in this gene that ignores DNA damage. And so a lot of cancers don't respond to radiation therapy because they've lost this ability to trigger apoptosis through that checkpoint. Now, chemotherapy, the name itself kind of tells you what it does. It's chemicals. Now, when we say chemicals or chemotherapy, there is a huge spectrum. When we say chemotherapy, we don't just mean one type of chemical. There are hundreds of chemicals out there that are designed to kill cancer. So what is the target of chemotherapy? Well, this is where we come back to what we discussed in the last lecture. Some chemotherapy drugs prevent the DNA from being replicated, and therefore the cell can't divide. Some chemotherapy drugs prevent the mitotic spindle from forming, so therefore they can't finish mitosis and the cells die, and so on and so forth. So the targets of chemotherapy are the genes that are necessary to initiate the cell cycle. They're essentially preventing DNA replication, preventing the things from undergoing mitosis, and the cells therefore cannot divide. So that's usually the target for chemotherapy drugs. They kill cells that are metastasized, and the distribution of chemotherapy can vary, but usually it's done intravenously through the bloodstream so that it reaches your whole body. This is done typically when you have cells that have metastasized, because you don't know where they've all spread to, and so you basically have to flood the entire cardiovascular system with this chemotherapy drug to try to kill any of these loose cells that are going all through your fluid system. And it's a very serious problem when a cell has metastasized. So chemotherapy is typically a chemical agent that's used to block the cell cycle and prevent the cells from growing and ultimately causing them to die. Yeah?
Is there a way that they can take out the chemicals after they run, or do they just put them in and they just have to wait till they lose their effect?
Well, some therapies, usually the body will detoxify, your liver will break them down, and then you'll get rid of them through your urinary and digestive system. A lot of drugs, in fact, I worked at a pharmaceutical company that was developing a drug that was designed to boost the immune system after chemotherapy. And so usually they'll do a round of chemo, and then they'll treat someone with a drug to try to restore the healthy cells and replenish their immune system and get their body back to normal before another round of chemotherapy. So yeah, the body will naturally eliminate the chemotherapy drugs that are going through the liver and going through the cycle, and then you'll go through another round. So usually people are given a dose, just like radiation therapy, they're given a dose. Now radiation therapy, there's a number of ways in which you can distribute it as well, but ultimately there's only one thing radiation therapy does. Damages the DNA to induce apoptosis. Chemotherapy essentially attacks the proteins and enzymes that are involved in mitosis and cell division, even DNA replication. So the last concept we're going to cover today are just some of the general characteristics of cancer cells. And then when we come back on Thursday, we'll really dive into the genetics of how cells regulate the cell cycle, where they have these checks and balances, where they, keep each other in check. Kind of how our government was meant to be, but is not. You could say it's cancers, but that's more on a political issue. All right, so cancer cells are very different from normal cells. Okay? Now, doctors who specialize in cancer cells are called what? Oncologists. Now, what do oncologists look for when they look at tissues? When you take a biopsy, and they take a tissue sample, and they're looking for cancer. What is it that they're looking for? Well, in the past, we mainly just looked at the cells, but now we do a lot of things to be able to test the genetics. Remember biotechnology, microarrays, where we look at hundreds of genes? That's another thing that's used today to profile the cancer cells to look at what genes might be going wrong. Now, putting that aside, let's just look at some of the general characteristics of cancer cells when we would call them cancerous. Number one, because they devote most of their time to growing and not doing what they were designed to do from their tissue of origin, they stop behaving the way that they should. So they also start looking differently. You can actually tell the difference between that. Now, this is a very high magnification. Doctors, when they look at cancer cells, typically don't see this much of a difference. But ultimately, they look through a light microscope, and they can see a different, what we call morphology or shape in the cells. It's not as obvious as this picture. If it were, it'd be way easier than it is today. But they're specially trained to look under the microscope, and they see that the cells have a different morphology. They look differently. Okay, we call that non-specialized. So what does that mean? Well, your skin behaves like skin because it produces the proteins that skin needs to behave like skin. When the cells start becoming cancerous, they devote less time to making those proteins and more time into growth, and therefore they become non-specialized. They look differently, they behave differently, they're not skin anymore. They're not that cell type anymore, they're now cancerous. Let's jump down here to the middle, this one, and then we'll just call it a day. Lacking contact inhibition. When cells become cancerous, they lose one of the inherent abilities that normal cells have, which is what we call contact inhibition. So what's contact inhibition? Well, think about it. When you cut your skin and your skin starts regrowing, there comes a point where the tissue has been repaired and you've packed your cells together. Normal cells, when they start having this contact with other cells, are like, oh, hey, we're done. We finished the repair. We're all back together again, and there's signals that tell them to stop growing. Well, when cancer cells get together, they're just like, make some more room for the next guy. They ultimately don't care about that contact, and this is what leads to the tumor mass, because it doesn't stop at its normal repair. It just keeps growing and growing and growing as much as it possibly can. Basics. So, since cancer comes from your own cells, many of the things you're going to see here are actually normal processes that are just taken to the extreme that are bad when cancer uses these things that are naturally occurring. And in some cases, they pretty much do the opposite as well. So you'll see both of those today. So the first one that we talked about specifically, and that is associated with formation of tumor masses, is the contact inhibition. Normal cells, when they repair tissues, like you get a cut in your skin and they start regenerating your skin, when they start touching one another and coming in contact with one another and filling up that gap and forming the scar tissue and whatnot, they essentially generate what we call contact inhibition, which is a way for them to signal to each other that the repair is done, they need to stop growing. So it's a way to turn off the accelerated cell growth. Well, cancer cells pretty much lack this contact inhibition, which is why when they form those tumor masses, they just keep growing and growing and growing without any concern with how big or how close they are to one another. They don't respond to this type of contact inhibition. So that's a big problem. That's why tumors essentially form. Another one is because they're devoting most of their time to growing, and they stop really producing a lot of the proteins that they were originally designed to do before they became cancerous, then they look differently, they behave differently, and we call them non-specialized. So you have about 256 different types of cells in your body, each one with a specific job. And when they're doing that job, we call them specialized cells. So pretty much each one of your cells has some respective job which they're designed to do. Skin behaves like skin, liver behaves like liver, and whatnot. But cancer cells pretty much look differently because they behave differently, because they stop doing the things that they're supposed to be doing, and we call that non-specialized. Okay? Another problem is that they can actually reinforce each other to increase cell growth. So as the tumor starts growing, the cells can say, hey, we're all having a party here, let's just get more in, and start causing them to divide even more. So it propagates itself. It kind of starts growing exponentially, and this is why you want to catch it pretty early, because of this positive feedback loop that can be generated by the cells as they increase in their overall mass. Now, one of the issues with cancer is it's not like normal cells where they have a finite cell division. Now, what I mean by that is normally when cells divide, they have a pre-established number of cell divisions, usually about 50, before they stop. And the reason why they stop is because at the ends of the chromosomes, remember we talked about the sister chromatids and whatnot, well, at the ends of the chromosomes, there are these protective DNA regions called telomeres. As the cells undergo cell division, these telomeres get shorter and shorter until the cells, once the telomeres are gone, the cells cannot replicate the DNA anymore. So they can't divide. The problem is with cancer cells, because they usually come from our adult stem cells retain the ability to divide indefinitely because they have an enzyme active called telomerase. Telomerase is an enzyme that essentially rebuilds the telomeres every time they get shorter. So they don't have a shelf life. They don't have the limited number of cell divisions. And that's why it's so problematic, because the cancer cells, having stemmed from these adult stem cells, have that ability to divide indefinitely because they have this enzyme telomerase active. Okay, so that's a problem. You have to kill them. They can't just wait it out and say, well, they'll eventually stop growing. No, they won't. Now, this is not true for all cancer, but since it is over 50% of cancer that is diagnosed today has this characteristic that I make mention of it. Over 50% of the diagnosed cancers have lost the ability to respond to radiation therapy, primarily because of a mutation in the gene that triggers apoptosis when the DNA is damaged. So that's one of the reasons why a good number of cancer cells, now not all, otherwise we would never use radiation therapy again, but a good number of them won't respond to DNA damage to induce apoptosis because of genetic mutations that are pretty ubiquitous in the human population. And then, here's the final thing that is a common characteristic of your normal cells, but is used to, is problematic when cancer cells do it. Your cells automatically regulate the flow of blood around the tissue so that they can get the nutrients that they need. We call this angiogenesis. So angiogenesis is essentially formation of new blood vessels. We're not talking about arteries and veins. We're talking about the smallest blood vessels that carry nutrients to the individual cells. Those are capillaries. So capillaries are only one cell thick, meaning one red blood cell can go through at a time. Well, a lot of times they'll get clogged, they'll get stuck, they'll get smashed together and get blocked. Kind of like if you have a little stream or a river and it gets blocked, it's going to redirect around that blockage. Well, unlike a river, where it can just kind of navigate its new way, you were dealing with actual blood vessels here. The cells, when they start getting starved for nutrients, secrete growth factors like VEGF, it's vascular endothelial growth factor. They secrete this hormone, and the cells essentially say, okay, let's form new capillaries around this blockage so that the cells can get the nutrients that they need. The problem with this is, as the tumor grows, it's also able to stimulate angiogenesis to increase the blood flow to all of the cells so that they get those nutrients that they need. And that becomes problematic, especially when you don't want the cancer to grow as fast as it possibly can. A lot of times we use drugs called angiostatins and endostatins, which essentially block these signals and prevent the cancer from forming new capillaries that surround the tumor mass. Sometimes when a cell gets infected by a virus or whatnot, you have immune cells, cytotoxic T cells that will essentially punch holes in your cells and tell them to die. There are these little receptors on your cells called MHC class 1 receptors that when they get infected with the virus or something like that, they'll present this portion of it to these cells, and when the cells recognize that, it's essentially the cell saying, kill me, So the T cell comes in and releases certain apoptotic enzymes that ultimately trigger cell death. So sometimes it's not the cell itself that triggers cell death, but your immune system. And this is also where it comes into play when cells become cancerous. They also can present these signals to your immune system, and as your immune system is monitoring all the cells in your body and it comes across one of these, it's like, oh, this needs to die because it's saying, hey, I've got problems, kill me. Okay? So it still is apoptosis, but it's triggered by another cell, typically your immune cells. So that's why a lot of times too people can have cancer cells, but their immune system takes care of it. Our immune system is designed to take care of it. What happens is sometimes the cancer cells don't present this tag on the surface of its cell membrane, and the T cells just overlook it. It doesn't bother with it. Okay, now let's talk about some of the genetic causes. Now, not all cancer is caused by genetics, but that's what we're mainly going to talk about today. There are many things that can cause cancer. In fact, they've shown that just taking cells out of the environment that they normally are in and putting them into a new environment can all of a sudden tell them to become cancerous. They're under a different set of hormones or signals or whatnot from adjacent cells, and they can become cancerous if they're in the wrong tissue. So we're not going to go through all of the things that can cause cancer, but we are going to talk about the genes, especially genetics, mutations that ultimately can cause cancer. So just be aware of that not all cancer is caused by mutations in the genes, but that's what we focus on a lot of the time because that's what's quantifiable. But they're finding more and more reasons of cells becoming cancerous that are not genetic mutations. All right. Sometimes a viral infection can actually cause the cells to become cancerous, and it's not a mutation of the genes, but it is caused by the virus infecting the cells and causing all sorts of problems. So in fact, we've used that same process to immortalize cell lines for research, where we actually infect them with a particular virus that essentially makes them a cancer cell, allows them to divide indefinitely. So we've seen what nature can do when we study it, and we do it for a number of different research purposes. Okay, now here's where we get into the checks and balances of the cell cycle. Remember I told you that the cell regulates its growth and death by certain groups of genes. Now we're not going to talk about any one gene in particular. These are actually groups of genes. Kind of like if we look at Congress and our judicial system and whatnot, they're made up of a lot of different people all forming one large group. And they're supposed to have checks and balances with one another. And then we have our executive branch, which is screwing everything up. They've been screwing it up for years, so I don't have any particular beef against one president or the other. But anyway, so ultimately, the two groups essentially do opposing things. They keep each other in balance. So the first group of genes we call proto-oncogenes. Now, these genes are specifically designed to accelerate cell growth and stop apoptosis when your cell needs to repair. When your body needs to repair itself, these are the genes that ramp up mitosis. These are the ones that be like, hey, you guys got to repair and grow faster because we've got a break in the integument in your skin and whatnot. So the proto-oncogenes are good genes because they're designed to accelerate cell growth when it's appropriate. And when cells are damaged, sometimes the adjacent inflammatory response can cause further cell death. And so one of the things these genes do is prevent apoptosis, because you don't want cells dying when you want cells growing. You want to turn on one and turn off the other. Well, when these genes become mutated, we don't call them proto-oncogenes anymore. We call them oncogenes. And again, what are doctors who specialize in cancer called? Oncologists. That's where they get their name from. So oncologists study oncogenes and many other things, but that's what we give the name for those who study cancer and treat cancer, our oncologists. Now, oncogenes, when they're mutated, they essentially are on all the time. And that's a problem, because when you're promoting the cell cycle constantly, that's what causes cancer. Now, one gene by itself is not enough to cause cancer. And that's why we have these checks and balances. So on the other end of the spectrum, this other group of genes we have are called tumor suppressor genes. Well, guess what? The name tells you what they do. They suppress the formation of tumors. How do they do that? They do the opposite of proto-oncogenes. So whereas proto-oncogenes promote the cell cycle and stimulate apoptosis, prevent apoptosis, tumor suppressor genes inhibit the cell cycle and they stimulate apoptosis. So they essentially do the opposite. So if one gene starts going out of control, the other genes can rein it in. Okay? So no one gene really can cause cancer. And this is the reason why it's not as prevalent as it could be, because, yeah, you can have one thing out of control, but everything else reins it in. It's when the genes that are supposed to rein it in, when they get mutated, now you've got a synergistic problem. Because now these genes are telling the cells to grow uncontrollably, and these aren't stopping them from doing so. Now, what's nice is that you have two of every type of gene, which means that you need two mutations in both of your genes to ultimately get this combination to cause cancer. Again, more protective nature of our chromosomes and of our genes. So by having two of every gene. The reason why we usually focus on oncogenes with cancer is because it only requires one of your genes to cause a problem for the oncogenes. But it requires two problems in your tumor suppressor genes to cause a problem. So that's the checks and balances that's going on here. Okay? Now, these are the ones, the tumor suppressor genes are the ones that step in once the repair is done and shut the proto-oncogenes off. So during normal repair, you cut your skin, the proto-oncogenes ramp up the cell cycle, and then when contact inhibition comes into play, that's when the tumor suppressor genes jump into play and say, hey, you're done. And if you have too many cells, it'll stimulate apoptosis. These are also the ones that trigger apoptosis through radiation therapy. So there's a common mutation in a very particular tumor suppressor gene that is found in over 50% of the cancers that are being diagnosed today because it gets inherited. So, like I said, one mutation is not enough. You've got to have the mutation in the tumor suppressor genes that would normally rein this oncogene in, and when you get that combination, then you get cancer. And this is why when you inherit these mutated genes, you end up having a predisposition to cancer. Okay? So let's talk about that. Later on, we're going to talk about what we call dominant alleles and recessive alleles. Dominant alleles, you only need one mutation for them to cause a problem. Recessive alleles, you typically need two mutations for them to cause a problem, which is why tumor suppressor genes are typically recessive and oncogenes are typically dominant. But, like I said, no one gene can cause cancer. However, you can pass an oncogene on, and you can pass a tumor suppressor gene on, a mutated tumor suppressor gene on. And that just makes you one step closer to having those cells have those issues because of the combination of different genetics. Now, what are some of these? These are the ones that they typically test for when you go and get a blood sample from, say, Myriad Genetics. They specialize in ovarian and breast cancer and things of that sort. One of the genes that they look at is called the BRCA gene. So the BRCA gene is one of those that's pretty prevalent and inherited, and it's an oncogene that gets passed on. So if you have a history of breast cancer in your family, from your grandmother to your mother and whatnot, then more than likely you might have that oncogene, which makes you that much closer to getting breast cancer. Now, some genes are found in all cells. Others are only expressed in various tissues. For example, RB or retinoblastoma. This is what causes eye cancer. This is one of the oncogenes that can cause eye cancer. There's other genes like the P53. This is that tumor suppressor gene. this is found in all cells. So if you have this mutation, this makes it possible that you can get any type of cancer, really. So it depends upon the gene and where it's expressed that ultimately determines what your probability of getting cancer in that type of cell is. And it's very easy to do this. In fact, this is where DNA sequencing comes into play. That you go in, they take a blood sample, and they'll run some genetic tests to screen and look at any mutations by sequencing your DNA in these specific locations where these genes are found, and they'll look for any mutations. And they might say, yeah, you have a mutation, but it's a silent mutation. It basically doesn't change the protein. You're good. You don't have a problem. But in other cases, they may say, yep, this one is one that we see a lot. It definitely gives you a predisposition. You could get checks more often and whatnot. So this is where profiling your DNA can really be important, especially if it runs in the family. A lot of times people come up afterwards and ask, what was the name of that company? It's called Myriad Genetics. It's up in Salt Lake. A lot of times your insurance company will pay for it because they'd rather prevent cancer than pay for it after the fact. So this is a place that specializes in DNA sequencing and things of that sort.